Today, I'll be talking about how age-related microbiome metabolites can alter RNA splicing and chromatin accessibility in the brain. On the screen, there's a link to our recent preprint on this topic. So over the past decade or so, it's become clear that the composition of the microbiome, the gut microbiome, changes with age. So this brings up a really interesting question. Do age-related changes of the gut microbiome impact brain function, and if so, how? So there are many ways to think about this question, but we thought about it through the lens of blood metabolites. It's now known that the microbiome modulates levels of blood metabolites. So here, for example, are some data that show that. Here in this graph, each dot represents the serum metabolome of a different mouse, either a germ-free mouse in yellow, these mice lack a microbiome, versus the conventionally raised mice in blue, which have a microbiome. And you can see a clear separation between the serum metabolomes of the germ-free mice versus the conventionally raised mice. So given this background, we hypothesized that microbiome aging affects the brain by altering the blood metabolome. Specifically, altered gut microbiome composition during aging necessarily alters the blood levels of micromodulated metabolites in the bloodstream. We hypothesized that these altered blood levels would alter the function of the brain's blood vessels and perhaps also of the surrounding tissue. Given this hypothesis, the goal of our project was to systematically characterize how blood metabolites linked to aging and the microbiome affect the brain. Our project had three parts. The first part was a computational portion where we mined public datasets to define a relevant panel of 30 blood metabolites to investigate. Next, we conducted an in vitro transcriptomic screen examining how the 30 chosen metabolites affect gene expression in human brain endothelial cells. Finally, we conducted an in vivo portion of the project where we examined how 4 promising metabolites from the screen affect brain cells and behavior in mice. So first, we'll talk about the computational portion. So here, we used public data sets to determine a panel of 30 metabolites to study, each of which satisfy the following 2 criteria. First, their blood levels are tightly linked to microbiome composition based on data from germ-free mice and over 8,000 humans. Second, their blood levels change with age in at least one large human cohort. So then we moved on to the in vitro transcriptomic screen, examining how the 30 metabolites we chose affect gene expression in human brain endothelial cells. In this graph, you'll see 12 metabolites that had a significant impact on the expression of genes in KEGG pathways linked to Alzheimer's disease and/or Parkinson's disease. On the x-axis are the 12 metabolites, and the y-axis shows the number of genes in these pathways that they perturb. Based on the yellow bars, you can see that each of these metabolites perturbed at least 5 genes in the KEGG pathways linked to Alzheimer's disease and/or Parkinson's disease. The affected genes converged on processes including energy metabolism, proteasome-mediated protein degradation, and inflammatory signaling, whose disruptions are central to neurodegenerative disease. From these 12 metabolites of interest, we then prioritized 4 for further investigation in vivo, and these are shown on screen: IAA, GDCA, TMAO, and PA. We used several criteria to narrow down the metabolites, and these 4 metabolites share several features. First of all, they have all been previously associated with Alzheimer's, Parkinson's, or impaired cognition in human cohorts. Second, they are all strongly associated with production or regulation by the gut microbiome. And finally, they have all been found in human cerebrospinal fluid or brain. So that brings us to the in vivo portion of the project, where we examined how 4 promising metabolites from our screen, which were just shown, affect brain cells and behavior in mice. So one of the metabolites we studied was trimethylamine N-oxide, or TMAO. And to get at its impact on the brain, we collaborated with Sophia Shi and Daniel Richard, from the labs of Tony Weisskore and Anne Brunet at Stanford. So we injected TMAO into the mice and looked at its effect on gene expression in the mouse brain. In this graph, each dot represents a different gene, and the x-axis represents the log 2-fold change in the expression of that gene in TMAO-treated brains versus control brains. So the purple dots are all the upregulated genes, and the teal dots are all the downregulated genes. And one thing that stood out to us was that many of the strongly downregulated genes by TMAO were actually splicing factors. And when we dug deeper, we saw that there were actually 44 differential splicing events between the brains of TMAO-treated animals versus controls, including differential splicing of the gene KMT2B, whose aberrant splicing has previously been associated with dystonia, which can occur either independently or as a symptom of Parkinson's disease. This is the first demonstration to our knowledge that specific microbiome-derived metabolites can alter host splicing patterns, whether in the brain or in any other host organ. And this is intriguing to us given the known dysregulation of splicing patterns in neurodegeneration. It is possible that microbial metabolites like TMAO might contribute. Another metabolite we examined was this microbiome-derived metabolite glycodeoxycholic acid, or GDCA. Based on a series of upstream experiments that I'm not showing due to the sake of time, we decided that we should do a TAC-seq on the brains of GDCA-treated animals versus control animals. In this plot, each dot represents a different genomic region in the brain. The purple dots represent genomic regions that became more accessible upon GDCA treatment. By contrast, the teal dots represent genomic regions that became less accessible upon GDCA treatment. And what you can see is that GDCA made hundreds of genomic regions in the brain less accessible. And this was intriguing because a more open chromatin state is generally associated with improved cognitive function, in stark contrast to what we observe here. So based on these findings, we wanted to see that due to this closed chromatin state, can we observe any behavioral changes in GDCA-treated animals? Here are some data showing mouse activity within a maze. So here on the x-axis is time that the mice spent in the maze, and on the y-axis is the distance that they moved per minute. The teal line represents vehicle-treated mice, the purple line represents mice treated with a low dose of GDCA, and the navy line represents mice treated with a high dose of GDCA. And you can see here a dose-dependent reduction in mouse mobility upon GDCA treatment. So to summarize, we first identified 30 microbe-associated metabolites whose levels change with age in human blood. Then we saw that in human brain endothelial cells, 12 of these metabolites regulate at least 5 genes relevant to Alzheimer's and Parkinson's, and we selected 4 for in vivo study. We saw that TMAO and GDCA induce significant molecular changes in the mouse brain, including impacts on splicing and chromatin accessibility, whose dysregulation is implicated in brain aging and related neurodegeneration. And finally, we saw that acute exposure to GDCA via oral gavage reduces mouse mobility. So the key takeaways here are that microbiome aging might substantially contribute to brain aging through impacts on the blood metabolome, and therefore that targeting the microbiome and its metabolites may offer neurological benefits during aging. Finally, the framework we describe, starting from metabolomics data analysis, going to an in vitro transcriptomic screen, and concluding with in vivo studies of host organs and behavior, can be applied to identify the microbiome metabolites relevant to any host pathology of interest. Thank you so much, and I'm excited to take questions during the Q&A session later.